Hello friends, I am Dr. Deepika Malik. Today's topic of discussion is industrial production of glutamic acid. Glutamic acid or its ionic form glutamate are underrated good dietary amino acids found in most high protein foods. The history of the first amino acid production dates back to 1908 when Dr. K. Ikeda, a chemist in Japan, isolated glutamic acid from kelp, a marine alga, after acid hydrolysis and fractionation. The breakthrough in the production of monosodium glutamate, that is sodium salt of glutamic acid or glutamate, was the isolation of a bacterium Corynebacterium glutamicum. The successful commercialization of monosodium glutamate with this bacterium provided a big boost for amino acid production. Since then, monosodium glutamate has been widely used in both the food industry and by the general public as a flavor enhancing agent. Glutamic acid commercial production by microbial fermentation provides 90% of world's total demand and remaining 10% is met through chemical methods. Now coming to the definition of glutamic acid is a non-essential amino acid that forms proteins. The amino acid has a chemical formula of C5H9NO4. Glutamic acid has the normal amino acid structure of an amino acid, that is an amine group, central carbon, carboxylic acid and an R group. In the case of glutamic acid, the R group is C3H5O2. This group is polar in nature. Polar means that this molecule is slightly positively charged on one side and slightly negative on the other. Now let us see some properties of glutamic acid. The major form of glutamic acid formed after fermentation is L-glutamic acid. It has a molecular weight of 147.13 gram per mole. It appears colorless to white, it is odorless, and it is sparingly soluble in water and insoluble in ethanol or ether. Its pH ranges between 3 and 3.5. Now coming to the uses of glutamic acid is widely used in the production of monosodium glutamate, which is commonly known as the seasoning salt. The world production of glutamic acid is about 8 lakh tons per year. Monosodium glutamate is condiment and flavor enhancing agent. It finds its greatest use as a common ingredient in convenient foodstuffs. It is used as a nutritional supplement. In cosmetics, it is used in treatment of hair loss and in preventing aging. Now let us see the biosynthesis of glutamic acid. The raw material which is provided during fermentation is broken down into fermentable sugar, that is glucose. Now this glucose is broken down by glutamic acid producing microorganism through the glycolysis pathway, and the end product, that is pyruvate, is channeled into the tricarboxylic acid cycle. As you can see in the diagram, this pyruvate is converted into acetyl coenzyme A, which enters the TCA cycle. This acetyl coenzyme is converted into citrate, isocitrate and finally into alpha-ketoglutarate. This alpha-ketoglutarate is the key precursor of glutamic acid. Alpha-ketoglutarate is converted into L-glutamic acid through reductive amination with free ammonium ions. The step is catalyzed by the NADP-dependent glutamate dehydrogenase. Now let us understand the steps involved in the regulation of glutamic acid biosynthesis. The knowledge of these regulation steps help us to develop improved strains of the microorganism for the enhanced glutamic acid production. For increasing the yield of glutamic acid, a continuous supply of citric acid cycle metabolites is required. Corynebacterium glutamicum has the enzymes phosphoenol pyruvate carboxylase for conversion of phosphoenol pyruvate and pyruvate carboxylase for conversion of pyruvate to efficiently produce oxaloacetate, which is a TCA intermediate, while the synthesis of glutamic acid occurs. Another key enzyme that can facilitate optimal production of glutamic acid is alpha-ketoglutarate dehydrogenase of citric acid cycle. Its activity has to be substantially low for good synthesis of glutamic acid, as is the case in Corynebacterium glutamicum. As you can see in the figure in the previous slide, that alpha-ketoglutarate is acted upon by the enzyme alpha-ketoglutarate dehydrogenase to form succinyl coenzyme A. Now we want our alpha-ketoglutarate to form glutamate, for which the activity of alpha-ketoglutarate dehydrogenase enzyme has to be decreased and the activity of the enzyme glutamate dehydrogenase has to be increased for the formation of glutamate. Further, exposing the cells to antibiotics like penicillin and surfactants 2040 reduces the activity of alpha-ketoglutarate dehydrogenase while glutamate dehydrogenase activity remains unaltered. By this way, formation of alpha-ketoglutarate to succinyl coenzyme A via citric acid cycle can be minimized, while the formation of alpha-ketoglutarate to glutamic acid is made maximum possible. Again, you can refer to the figure on the previous slide for better understanding of these points. Now let us understand the role of biotin, which is a water-soluble vitamin, in glutamic acid production. Glutamic acid production is greatest when biotin is limiting. That is, limiting amounts of biotin restrict cellular synthesis and induce synthesis of high yields of extracellular L-glutamic acid. When biotin is sufficient or optimal, bacterial growth is luxuriant and glutamic acid formation is negligible. So basically, high amount of biotin will favor bacterial growth, whereas in low concentration, formation of glutamic acid will be favored. The optimal level of biotin is 0.5 mg per gram of dry cells. With higher amounts, glutamic acid production falls. That is, for efficient production of glutamic acid, the concentration of biotin should be below this value. An increase in permeability of the cell is necessary so as to permit the outward diffusion of glutamic acid, which is essential for high glutamic acid production. This increased permeability to the acid can be achieved by ensuring biotin deficiency in the medium. Biotin is an essential cofactor for the biosynthesis of fatty acids. Due to a limited supply or deficiency of biotin, fatty acid biosynthesis and consequently phospholipid synthesis is drastically reduced. As a result, membrane formation, that is protein phospholipid complex, is defective, which alters permeability for an increased export of intracellular glutamic acid. So to sum up, deficiency of biotin affects the proper formation of cell membrane, thus affecting the membrane's permeability and allowing the secretion of glutamic acid outside the cell. Now coming to the raw materials, carbon sources used can be refined, which includes glucose, sucrose, fructose or maltose. These are high cost carbon sources. Another category is unrefined carbohydrate sources such as sugar beet molasses, sugarcane molasses, etc. Another important component is the nitrogen source. The concentration of ammonia is very crucial for converting carbon source to glutamic acid. However, high concentration of ammonia inhibits the growth of the organisms. In the beginning of fermentation, ammonium salts and a low concentration of ammonia are added. During the course of fermentation, ammonia in aqueous solution is continuously fed. In this way, pH can be controlled besides continuous supply of nitrogen source. Sometimes, urea is also used as a nitrogen source, since glutamic acid production bacteria possess urease that can split urea and release ammonia. Now, selection of industrially important microorganisms depend upon the following properties. It should have the capability of producing spores or can be easily inoculated. It should grow rapidly on a large scale in inexpensive medium. It should produce desired product quickly. It should not be pathogenic and it should be amenable to genetic manipulations. The glutamic acid is produced through the fermentation process. Major organisms used are Corynebacterium species, which includes Corynebacterium glutamicum and Corynebacterium lilium. Brevibacterium species like Brevibacterium divaricatum and Brevibacterium alanicum. Microbacterium species like Microbacterium flavum var glutamicum, or Arthrobacter species like Arthrobacter globiformis or Arthrobacter aminofaciens. The preferred organism among all is Corynebacterium glutamicum. The properties common to the glutamic acid bacteria are they are all gram-positive and non-motile, they require vitamin biotin to grow, and have a high activity of glutamate dehydrogenase and a low activity of alpha-ketoglutarate dehydrogenase, which means high activity of glutamate dehydrogenase leads to the formation of glutamic acid and a low activity of alpha-ketoglutarate dehydrogenase leads to less formation of alpha-ketoglutarate to succinyl coenzyme A. Now after the selection of the raw material and the microorganism, next is the fermentation process. Various fermentation approaches for the production of glutamic acid include batch fermentation. In batch cultivation, all the nutrients are added at once at the beginning of the fermentation. except for ammonia, which is added intermittently to help adjust the pH, and fermentation continues until sugar is exhausted. Second approach can be fed batch fermentation. That is, the fermenter is only partially filled with medium and additional nutrients are added either intermittently or continuously until an optimum yield is obtained. And the last approach can be continuous fermentation, in which addition of substrate and product removal is done simultaneously. Among all the three approaches, the fed batch appears preferable for the following reason. Most amino acids production require high sugar concentration of up to 10%. If all were added immediately, acid would be quickly produced, which will inhibit the growth of the microorganisms and hence reduce the yield. Fermentation completes at 38 degrees Celsius with pH 7.8 within 2 to 4 days, and at the end of the fermentation, the broth contains glutamic acid in the form of its ammonium salt. Now, after the fermentation is complete, the next step is the product recovery. For cell separation, centrifugation and filtration are done. Centrifugation is a common method used in industry, can be operated in semi-continuous or continuous basis. Poor centrifugation can be improved by adding flocculation agent. This agent will neutralize the anionic charges on the surface of microbial cells. Upon neutralization, these microbial cells join together and settle down at the bottom. Next is filtration. Filtration is widely used in industry and can be improved by using filter aids. This separates unnecessary cell debris and impurities. After that, ion exchange is used for the extraction and purification of amino acids from the fermentation broth on the basis of charge of the solution. Glutamic acid anions get bound to the resin and ammonia is released. This ammonia can be recovered via distillation and reused in the fermentation. After that, the glutamic acid bound to the resins is eluted with sodium hydroxide. With sodium hydroxide, glutamic acid forms monosodium glutamate, which can be purified by passing through anion exchanger. The monosodium glutamate is cleaned by using active carbon. After treatment with sodium hydroxide, the next step is evaporation and crystallization. The clean monosodium glutamate solution is concentrated by heating and the monosodium glutamate crystals is formed. The crystal produced are dried with hot air in a closed system. Then the crystal is packaged in the packaging and ready to be sold. The next slide shows a schematic diagram for glutamic acid production. Now let us have a quick review for the production of glutamic acid. First, the raw materials are prepared in the dissolving tank. They are sterilized and its pH is set before entering the fermenter. After the raw materials has reached the fermenter, it is inoculated with the bacteria. After fermentation, cells are removed by centrifugation and filtration. The broth is then subjected to ion exchange resin where glutamic acid gets bound, which is eluted further by sodium hydroxide. This generates monosodium glutamic acid, which is further purified by passing through an ion exchanger. After that, evaporation is done and then crystallization is done where the crystals of monosodium glutamate are formed. The crystals are finally dried and packaged for selling. With this, we wind up the topic of industrial production of glutamic acid. For any doubts and queries, you can contact me through the given email ID. Thank you.